and then complementary. And so complementary is a key thing too. What's in one strand is going to dictate the other. So if you have a C here, you got to have a G there. Okay? If you have an A here, you got to have a T there. Or if you're talking RNA, a U. And so when RNA folds up on itself, it's going to be anti-parallel and complementary to itself. Okay? If RNA, when we're talking DNA replication, we're going to break these hydrogen bonds, and the DNA polymerase is going to read this base and say, oh, I need to bring in a C. Then we're going to have a DNA polymerase over here reading this base and know, oh, I got to bring in a G. That's exactly what we're doing when we're talking about PCR, or Sanger sequencing. We're doing DNA replication in a tube. We're just synthesizing DNA. We're using DNA polymerase, and we're just giving the polymerase a template. And then rather than having primase design primers across the entire thing, we're just providing primers for what we want to amplify. Okay? And so that's what we're doing with PCRs. The primers we use are doing the same exact thing that the primers that primase is doing during DNA replication, only we're giving the primers. And so we're deciding where we want DNA polymerase to actually land and start synthesizing a piece of DNA. So that's what when we're doing PCR, or we're doing Sanger sequencing, or any of the other sequencing technologies. Ultimately what we're doing is DNA synthesis, which is all going to be based on this complementary base pairing and this semi-conservative nature of the way DNA replicates itself, and that we're able to use one strand to create another strand. Okay? And then also, this is where RNA transcription is coming in. So when we're going to separate these strands, we're going to have an RNA polymerase land on here, and it's going to know to bring in a G, and then it's going to move down, it's going to know to bring in a U. Okay? And so that's what RNA polymerase is going to use as a guide to be able to determine which nucleotide to bring into its growing strand of RNA. So DNA polymerase and synthesis is going to use this idea of complementary base pairing to dictate what the new strand is going to be created, what new strand is going to be created. RNA polymerase is going to use complementary base pairing to determine what nucleotides to incorporate into its growing RNA. And when this complementary base pairing is what we're using whenever we do any sort of hybridization. So if we're talking southern blots, northern blots, microarrays, fish, okay, all of these things, what we're using is hybridization. And so we're going to have a particular region of the genome that we're interested in, okay? Say, if you think back to the fish example, that's fluorescence in situ hybridization. And so what we're doing is our question is on these metaphase chromosomes, is the sequence that I'm interested in there, okay? It's just like a northern blot or a southern blot, presence or absence. Is the sequence there or not? And how we're able to determine whether the sequence there or not is if our probe hybridizes there, okay? And so if we make a probe, and so say this is a region of, say the example in your lecture notes is a 7q deletion. And so on chromosome 7q, it's a syndrome that you can get a little segment deleted, okay? And so individuals that have that deletion will have certain physical characteristics, and the clinical geneticists will go, hey, look at those characteristics. That looks like they might have 7q deletion. Okay? And so what they'll do is they'll do blood draw, they'll do chromosome prep, and put metaphase chromosomes on the slide, and they'll design a probe that's complementary to that region that's usually deleted. Okay? And so now you heat up your slide, that makes your chromosome single-stranded, you heat up your probe that you've made in the test tube using a polymerase, and then you just incorporate fluorescence into that probe. So wherever it hybridizes to, you can just see it through fluorescence. And you take your probe for that region of 7q, you heat that up, it becomes single-stranded, you heat up your slide, your metaphase chromosomes become single-stranded, put them together and let them cool. When it cools, these hydrogen bonds reform, and so wherever your sequence is that is complementary to your probe's going to hybridize to by complementary base pairing. Okay? That complementary base pairing is going to let that probe stick there, and you can just use the fluorescent microscope and look down and see whether or not you have a signal. That's what we're doing when we're doing a northern. That's what we're doing when we're doing a southern. Is in a southern, we're just chopping up the genome to make it smaller so it'll actually run through the gel. But all we're doing is spreading the genome out on a gel, taking a probe that's labeled with radiation, that's complementary to the sequence that we're interested in, and sticking it onto the gel transferred to a membrane, but sticking it on there. Wherever it sticks, it's going to stick by complementary base pairing, and then we'll detect where the radiation is. If no radiation is on that membrane, that means that sequence is not there. If radiation is on the membrane, that means the sequence stuck to the membrane by hybridization, and that our sequence is there because the complementary sequence is present on the actual membrane, okay? So we're able to detect presence or absence of these sequences based on actual complementary base pairing, okay? So whenever we're looking at hybridization, that's the general principle that we're using, is this idea of complementary base pairing. Whenever we're synthesizing DNA, we're using complementary base pairing and anti-parallel, just like we do, just like the genome or the cells do when it goes through the cell cycle and replicates the genome. That's what we're doing, only in a targeted way in a test tube, but we're doing exactly the same thing. So, now, we talked about the cell cycle way back, I think, on day 1. And it's important to revisit the cell cycle, because the cell cycle, most cells are going to divide over the course of their life. And where we kind of get the functional end of the cell cycle is, or where we kind of picked up talking about the cell cycle again is in the cancer topic, the cancer section. And so typically, remember, we have our cell cycle, we got G1, that's with the gap1 phase, and that's where the cell's just doing its thing. Then we hit this G1S checkpoint. We talked about how retinoblastoma contributes to the control of the G1S checkpoint. Before we proceed into the S phase, this is where we're going to be synthesizing our DNA. This is where those replication bubbles are. This is where the DNA synthesis is. We're copying our DNA. That's where our chromosome goes from one piece of DNA into 2 sister chromatids, okay? At the end of that, we go into the G2 phase, which is just another gap phase. The cell's being a cell. A heart cell's a heart cell, a brain cell's a brain cell, okay? Then we hit another checkpoint, the G2M checkpoint, where before proceeding into either mitosis or meiosis. The key thing to remember with the cell cycle is often with cancer, there are a couple of ways cancer can go wrong. We talked about the different types of genes that cancer go wrong. But often where the cancer genes are hidden are these checkpoints, because those checkpoints are where the cell takes a minute, takes stock of itself and says, am I intact? Is my genome intact? Do I have any chromosomes? Do I have any breaks? Do I have any misincorporated nucleotides? If you do, this is where the DNA repair machinery comes in and fixes things, is at these points. It's where the cell says, do I even have all the proteins and all the resources to be able to synthesize my genome completely, okay? And so these are really important, because if this is where errors are fixed, are at these checkpoints. And if we don't take time at these checkpoints to fix those errors, that's how we can accumulate mutations over time. And that's what the clonal evolution of cancer was really referring to, is that if you start blowing through these checkpoints, those rare little mutations that occur all the time, but are often fixed because we take the time to fix them, aren't given the chance to be fixed. And so now you're going to just keep accumulating mutations, and then which ultimately some of those mutations might hit other oncogenes or other tumor suppressor genes, which is going to increase the cell cycle even more, okay? And so we talked about the type of genes, so oncogenes, tumor suppressor genes, DNA repair genes contributing to cancer. So remember, oncogenes, that's like your gas pedal, that's stepping on the cell cycle, that's making it go faster. So if you increase amounts of oncogenes, well, that's how oncogenes have their effect. It's like stepping on the gas more. So you increase expression, so you have a translocation that brings an oncogene into a new region, and now it's being expressed too much. Or you have a segmental duplication that duplicates that region. Now you have 2 copies of your oncogene, and you're expressing twice as much. You're stepping on the gas more. Alternatively, we have tumor suppressor genes. These are the brakes in your cell cycle. This is what holds things up and how it slows it down and allows it to take that chance and take that time at these cell cycle checkpoints. And so if you lose your brakes in the car, so this is where you get a reduction of expression, is with the tumor suppressor genes. You're not stepping on your brakes, or you're losing your brakes. You get a deletion that deletes your good copy of retinoblastoma. Well, now you no longer have brakes, and you're going to blow through your G1S checkpoint. And so we talked about how the cell cycle and these mutations can ultimately contribute to cancer, and ultimately cancer is the uncontrolled cell cycle and the different types of genes that can control it. So, meiosis, mitosis. Y'all had it way back in high school. What's the earliest anybody had meiosis, mitosis? Where are they teaching it now, like 7th grade or something?
Yeah.
Really?
Yep.
Yeah. Wow, that makes me feel old. I didn't have it until like 10, when I was just got I think it's a little tiny piece of my biology book. So, but anyway, so meiosis and mitosis. You memorized it in 7th grade, but you probably didn't recognize the impact of it in 7th grade. Okay? And so a couple of things really to remember. Well, mitosis, we talked about it, that's just cell division. You duplicate your genome, you have sister chromatids, line your sister chromatids along the metaphase plate, split them up, you now have 2 copies of it. You have same endings, two cells with the same genome as what you started with. Okay? Meiosis, on the other hand, this is where all of the interesting stuff comes in, because remember, we're going to duplicate our genome, and then during prophase 1, this is where we're going to have our homologous chromosomes line up. Okay, we're going to start crossing over. The significance of that crossing over, we saw several times throughout the semester. This is what we're talking about in linkage. When we're talking about linkage and recombination frequencies, what we're talking about is the probability that 2 points are going to be exchanged on those homologous chromosomes, and that's going to be a function of how far apart they are. The further apart they are, the more likely they're going to be exchanged. The closer they are, the less likely they're going to be exchanged. Okay, and so that's what recombination frequency and linkage analysis ultimately give us, is because of this crossing over during prophase 1. So remember, if you have a 50% recombination frequency, well, that's the same as if it's just a 50-50 shot, as if you're just aligning independently assorting chromosomes like Mendel was studying. Well, 50% recombination, well, we don't know if they're linked. They could be linked, because they're so far apart we just can't tell whether that they're linked or that they're on separate chromosomes. But if it's less than 50%, we know that they're on the same chromosome, and the relative frequency in which they recombine is going to tell us the relative distance they are from each other. And so we have lots of information. That gives us lots of information, and we're able to do a lot with that. And then the other really important thing about that is it adds variation. It starts mixing things up. Your dad is from one particular lineage of people. Your mom's from another particular lineage of people. If we didn't have recombination, all of your dad's stuff would always stay as your dad's stuff, and it'd just stay intact. Well, now we have the opportunity to start switching and swapping. And so every time you have gamete formation, you start switching and swapping your maternal and paternal genomes from each other and start mixing up these different lineages. And so you add variation. We talk about in evolution how variation is good. It gives that population a lot like a buffer to be able to absorb changes in the environment. And so variation is a good thing, and that's what prophase ultimately is doing, is this recombination. But we're able to use that recombination in genetics to be able to start establishing relative position of loci on chromosomes. We also talked about metaphase and metaphase 1. In metaphase 1, this is where Mendel's stuff really comes down to, is really what Mendel was studying, was what's occurring at metaphase 1, of when 2 independently assorting loci, green and yellow peas versus wrinkled and round on different chromosomes, and how they assort with each other. And so remember, we have a 50-50 shot of the mother's chromosome going on one side and the dad's this way, or swapping, okay, for every chromosome. So again, this is going to add variation. This is an opportunity for gametes to not end up with all just your dad's chromosomes, but a combination of your dad's and your mom's, and some of them with mixed up prophase recombinations. Okay, so we can add variation, and then this is where we're going to be seeing the actual independent assortment, is what Mendel's work was showing. This is where we get our 3-to-1 phenotypic ratios in a monohybrid cross, our 1-to-2-to-1 genetic ratios in a monohybrid cross, our 9-to-3 to 3-to-1 in our dihybrid cross. All of that stuff is all because of this independent assortment of different chromosomes on the metaphase plate. So then we go into anaphase 1, and we split those homologous pairs. It's at the end of anaphase 1 that we're actually now haploid. We now have an N. Here we have 2N. We start with 2N. At the end of this is where we have N. And at the end, we end up with 4 chromosomes after going through another round of cell division. Analogous to metaphase, we have 4 cells that are haploid. Okay. So, again, this is a regular 9-to-3 to 3-to-1. And then remember, when we're talking Mendel, we talked a whole lot about Mendel's and extensions of Mendel's, and then when we're talking even quantitative genetics, what Mendel established is the way genes are inherited holds true all the time, whether you're talking about complex characters such as quantitative genetics, or whether you're talking simple qualitative characters like what he was studying. The inheritance always holds true because this is the way all gametes are formed. Okay, and so in extensions of Mendel, we talked about all variations of things, the lethal alleles in incomplete penetrance, variable expressivity, codon.